Our next speaker is Dr. José Aragon-Martin, who is the director of the Sonalee Laboratory of the Marfan Trust. For 20 years, the Marfan Trust has been referring supporters with eye problems to Moorfields Eye Hospital for expert care. Out of this mutual respect has grown a warm collaboration. So when Moorfields staff heard we were looking for a temporary laboratory, they offered Dr. Aragon-Martin a place to continue his research and teaching in the Institute of Ophthalmology beside Moorfields Hospital. There he's continued to study the genetic causes of dislocated lens, which occurs on its own and needs to be distinguished from Marfan syndrome. He's also written several papers and grant applications in order to continue our research into our main area of interest, further genetic causes of thoracic aortic aneurysm and dissection. Today, he's talking about progress in identification of ectopia lentis genes. Thank you, Jose.
Thank you. Yes, today I'm going to talk about the research we are doing in ectopia lentis. First, to say that the clinical features in Marfan syndrome can be seen in either the cardiovascular system with thoracic aortic aneurysm, mitral valve prolapse. Those are two examples. You can have also in the skeletal system affected with scoliosis or arachnodactyly. And also in the ocular system, you have the ectopia lentis or myopia. Again, these were just examples of the three systems from the many clinical features expressed in Marfan syndrome patients. In this talk, we are going to focus on the clinical features called ectopia lentis or EL. My picture here is a little bit more old. On the left-hand side, you have the picture of an eye where you can see the lens located in this position here. And this beautiful picture actually from 1979 is still amazing it's being with the zonular usha. Where did it go? There you go. Yeah, so the fibers are here seen what we call zonules or fibers, zonule fibers or zonules of zinc. And you can see here is the lens, and here you can see the iris. So yes, so the eye is a complex machine composed of many parts. The lens is one of them and is essential for focusing the light rays on the retina to turn light into vision. The lens does this by changing its shape with the help of the zonules of zinc, also known as the zonular fibers or zonules. We find the zonular fibers in the white fibers, these white fibers here. They come from the ciliary body, which is this region here, and they go to the equatorial part of the lens, which is this one here. So around the whole lens, we know that they appear to decrease with age. It has been suggested, it has been suggested, yes, in studies that the zonule are either formed, maintained, or both by ADAN-TSL4 protein. The zonular fibers are circular, cell-free structures that are composed primarily of fibrillin 1, which is made from the FBN1 gene. Although the zonular fibers are elastic structures that can be stretched to almost 4 times their original length without breaking, malformation of these fibers can lead gradually to lens dislocation or ectopia lentis, which is the partial or complete displacement of the lens from its space. This picture over here shows an eye where the lens has moved upwards because the zonular fibers are fragile or broken, and you can see the fibers here. The absence of traumatic injury implies that there must be a genetic component. EL can be found as dominant when one copy of the gene is affected, or recessive when both copies of the gene are affected. It has been shown to occur in isolation and as a part of a syndrome. While ADAN-TSL4 gene, which is this one here, has not yet been shown to have any cardiac involvement, FBN1 is linked to thoracic aortic aneurysm, clinical feature that is seen in many Marfan syndrome patients, and this can be fatal if not treated in advance. We are interested in the apparently isolated condition of EL. It will help us to, in our research approach, to focus first on those patients with ectopia lentis and no other clinical feature to discover new genes causing EL. But why are we interested in discovering new genes? Not much is known about the prevalence of EL, but a Danish study in 1998 gave a prevalence rate of 6.4 per 100,000 live births. In the same study, the etiology was established in 69% of these cases, but 31 were unknown. We have a cohort of 80 families where 40 families have an unknown etiology. Therefore, our cohort have 50% of families without a genetic diagnosis. So we started a pilot project with a cohort of 80 families, including 131 patients in total with isolated ectopia lentis, where the principal participant of each family was Sanger sequence with the two known isolated EL genes, the FEN1 and the ADAMTSL4. We found that 8 families were positive for FEN1 and 32 families were positive for ADAMTSL4. This makes sense since FEN1 and ADAMTSL4 are proteins found in the zonular fibers. In our experience, when EL patients are screened for these two genes, many are found who do not have a causative variant in their DNA sequence. This pilot project has reduced our cohort to 40 families that remain unexplained. Comparing our data with the Danish study in 1998, this could mean that either there must be new genes at play or that our approach was not sensitive enough to find the causative mutation. Therefore, further investigation is required to discover the molecular genetic basis. This is why we designed a new research approach. Our approach is to use NGS to discover new genes in EL using the cutting-edge technology called whole genome sequencing. This will allow us to interrogate regions that are not routinely studied when looking for mutations in the FEN1 gene, like intronic variants or large structural variants. As an example, here we have 2 pictures. This one is one of the pictures, and this one on the bottom is the second picture, showing the full intron region between exons 56 and 57 of the FEN1 gene. This blue box here is the first exon, the thing here, and the second exon is here for both of these patients. And the area in between is all here, is the intronic area. So the top picture is taken from the whole exome sequencing patient, and the bottom is from a whole genome sequencing patient. The continuous gray area here, we can see on the second picture on the bottom, it's all continuous here, is the coverage of the technique used. Notice that the coverage in the top picture, which are these 2 mountains here on the left and here on the right, is limited to the region between the exons and the start of the intron. So again, this is the exon here, and this area is the intronic area here. While on the bottom picture, yes, is more consistent and covers fully the whole intronic area. So it's all this area here is being covered by the technique. Oh yes, there are here some reads. But we think it's just other mistakes, since this area should be free from data from the whole exome sequencing. The difference might not be important at first sight. Here is an example of an intronic variant that affects further, that after further research was classified as causative mutation in a Marfan syndrome patient. This study is of the intronic variant was done in Italy by Fusco in 2019 using another technique called Sanger sequencing. And just to show you that here, I mean, there's a line here, but actually what it means is that this is the location where the intronic region is located, and that's why whole exome sequencing can also, both of the techniques can pick up the location. But the difference coverage between the whole exome sequencing and whole genome sequencing might increase the scope of the assay. Here, there's an example of an intronic variant, also known as deep intronic variant, that after further research was classified as causative mutation in a Marfan syndrome patient. The study of this intronic variant was done in Belgium by Gilles in 2014, after several assays were negative, including DHPLC, Sanger sequencing, MLPA, and whole exome sequencing. Finally, when they studied the cDNA of the FEN1 gene, the cDNA is a sequence of DNA without intronic information, they started to see the approximate location of the causative mutation. If the assay was done using whole genome sequencing, it would have been positive. You might ask yourself, why is it a mutation in the intronic causative? So this is a picture of the protein that forms the spliceosome. So all these proteins here are part of the spliceosome, and they bind to the DNA sequence. And this is over here an exon with the DNA sequence. And so here is the intronic region with some interesting DNA sequences here as well. So so some of them will bind into the exon sequence, and some will bind into the intronic sequence. So the spliceosome is a group of proteins that will use the intronic sequence to join exons from a gene to later produce the right protein. Mutations in the intronic areas of a gene are very important because they might change the sequence of the DNA that is required for the spliceosome to do its job. Using whole genome sequencing will also help to rule out non-ectopellentis genes. This table is a list of mutations of known EL genes from a patient. Each row is one mutation, and each mutation needs to be further analyzed to confirm it as causative or not. This step is imperative since we want to make sure that there is absolutely no causative mutation in a known gene, so we can continue to the next step, which is to discover new genes without any hesitation. To look at the known genes using the whole genome sequencing approach, we just need to extract those genes that we are interested in and study them. And since there are a few of them, this could be done quite fast. There's an online tool that creates lists of genes that are associated with a particular clinical condition, and it is called PanelAb. At the first look at the PanelAb, we realize that there is no list of EL genes there, but there is a panel related to eye disorders called structural eye disease, which contains many genes that might not be relevant to EL, but it can help us to create our own EL list of genes. One umbrella project from EL is to build a network of scientists to help us to create the first EL panel in this tool called PanelAb to help others in the right direction when it comes to research in EL. So after rolling out the known genes, it is time to search for new genes. To discover new genes in our cohort of 40 EL families, we can perform either trio or family studies. At the moment, we have 27 trio studies. One example of a trio family is over here on the bottom left picture. We have 5 small families like this one over here in the middle. And we have 8 large families. One of them is here on the right-hand side. If we screen our 40 families by whole genome sequencing to study the known genes, we can use the same data generated to study the new genes. So there shouldn't be no extra cost to do this step since we already have the data. This time, instead of extracting and studying selected genes, we can study all the genes in our body and filter out those that are not relevant to our study. We will compare affected members, and those genes found in all affected members will be selected. Then we will do gene interpretation studies. We will generate a list of candidate genes because we are expecting to have at least 20 of them. After this, we must perform family segregation studies. This is done by testing all members in the family to make sure that the variant we found in the preliminary studies is found only in the affected and absent in the unaffected patients. You can see on the predictor here, the circle ones, denoting is a female, and the square denotes is a male. Anything that is white is unaffected. Anything that is black is affected. And you can see that all the ones that are affected have the yes to go ahead, and then the one without, and the two without the that are affected with the no. To the note that this is a right approach. After performing successful family segregation studies, we must screen the rest of the EL cohort to rule out this gene. And therefore, if it is negative in patients from the EL cohort, these families will be included to look for a new gene. Our NGS research approach using whole genome sequencing is one of the most advanced techniques used to analyze mutation in DNA. I would like to reiterate that as far as we know, Sonalee Laboratory is the only lab in the UK that is working in finding genetic mutations that causes ectopellentis. Or please send me an email and let me know if you know of any other lab, because we would like to create a consortium to help each other on this matter. We have also helped the NHS by looking the genetic cause of the genetic cause in several EL families from the UK, which were negative for FBN1 gene, since ADAN-TSL4 gene is not routinely screened in NHS labs. Just as a fun fact, the NHS in St. George's came to Sonalee lab to ask for help to create a genetic test to screen Marfan patients, since we were the experts at that time. So we like to be very helpful. There has been a recent announcement last week from Illumina on whole genome sequencing. Just to say that Illumina is a company that creates the machines to run this kind of samples. They're announcing that the cost of running one genome will be about 200 pounds. The genome is the DNA that contains all the genes in your body. This is great news, since at present the cost of whole genome sequencing is less than 700 pounds, but less expensive than Sanger sequencing, which is still very expensive at a cost of about 900 pounds for sequencing just the FBN1 gene. Because we do the bioinformatics in-house, our expertise in this field of bioinformatics makes the Sonalee laboratory one of the most updated labs in the UK in molecular genetic detection and research. The data generated is huge, so it needs to be processed in special computers using the command line. These computers are called data scientist computers. We had to learn new computer languages like Unix and Perl to process the data, which was a little bit tough. Before I finish with my talk, I would like to say that we are always welcoming a volunteer hand. You can learn the skills that will shape your future career while helping us in the lab. If you are interested in a career in genetics, biology, or similar, we can help you to shape your laboratory skills. If you are going to work from home, we can accept A-levels or university students to help us with literature reviews and projects like genotype-phenotype correlations in Marfan syndrome. If you're going to learn bioinformatics or laboratory skills with us, we could prefer if you are at university level. Before I forget, yes, because of our express skills, we are attracting collaborators. We are creating a national group of experts in the matter to help each other to clarify the significance of the genetic basis of the clinical features seen in ectopilentis patients. And at the moment, it's made of Sonalee lab, which is Dr. Child and Dr. Aragon-Martin, myself, and several ophthalmologists from Moorfields Eyes Hospital and other hospitals around the UK. Here we have Professor Charteris over here, and then Mr. Chandra on the right-hand side here, who represents some of the UK collaborators. Also, we are building an international group on EL research, but at the moment, this is being discussed. One of our international collaborators is here on the bottom left, Dr. Don Sims from California. I would like to take this opportunity to say thank you to all the patients, families, volunteers, students, and sponsors, and especially to those who raise funds to help Sonalee Lab to continue what we love doing, which is to find the genetic cause of Marfan syndrome patients and in other related connective tissue conditions. So thank you very much.
José, we're very grateful and for your most erudite discussion. I'd like to point out this is a very practical project. As you mentioned, if we discover that it's just pure ectopielentis and no cardiac involvement, that's a great relief for the patient, I would imagine.
Well, I was going to say that we put everybody together in the pot, and then we study obviously the ones who are with no, what we think they don't have any other features but ectopielentis, but we put everybody together because it's cheaper for us to send the samples in a, I mean, right now it's 700 pounds to send them for whole genome sequencing, but if we send them in a group, then it's only perhaps 600 pounds. And if now it's gonna be 200 pounds, we are gonna be doing business. So yes, and the good thing about this is that if we send them for a screening, we don't have to waste a lot of money in other techniques, which we have already a technique that will allow us to look for many areas. I mean, there was one question I think before from somebody who was saying that if my kid is negative for the test, will he have Marfan syndrome? I'm not a clinician, so I cannot say anything, but I will say what technique have they used? Have they used the latest technique? And if they have used a particular technique, have they used this other technique? Because we have to make sure as well, it's not that you are going to be negative, but we want to make sure that we found the solution for the patient. So if it's Ehlers-Danlos gene, then we know that the cardiac involvement will be, we think anyway, that it's not going to happen there. So you have more chances to not to go to the doctor every year for checking. So yeah.
Right. I think we have time for one question. Victoria, is anybody asking a question?
I'm searching for raised hands to no avail. I can't see anything's emerged on the chat feed either.
Right. Well, I'd just like to make the point that Jose's tests also allow us to do appropriate genetic counseling, because Marfan syndrome is dominantly inherited with a high risk for each child, 50%. But if we find that the causative gene is for dislocated lens, these are usually inherited recessively, like cystic fibrosis. So usually there is no real risk for the offspring of someone who has dislocated lens, as long as they don't marry their first or second cousin. So well done, Jose.
Before I forget, I was going to say that we are having a database which we collect the data. And we don't delete anything. So in the future, if somebody comes to us, oh, my grandfather has the test with you guys, and I think I have it too. So then we know for sure what to test